Good morning, everybody. I'm Sandhya Jayasekara, and thank you for being here today. And today I'm going to share with you the findings of my master research work. And the title of the work is Characterization of the potent sorbitol dehydrogenase from a novel subsurface bacterium, Caldiatribacterium inferamans SIUC1, insights into structure and function. So first of all, I will give an overview of sorbitol dehydrogenases. So these are enzymes that catalyzes the interconversion of sorbitol into fructose. So the catalytic activity of SDHs needs the presence of certain cofactors, such as NAD+, NADP+. And these enzymes are found in most of the biological systems, including bacteria, yeast, plants, and animals, as well as in humans. And so they are categorized into a certain special group of enzymes called short-chain dehydrogenases or reductases. There are some evolutionarily conserved characteristics in this particular group of enzymes. That is, they have a zinc metal binding active site, and also they have this Rossmann fold, which is responsible for cofactor binding. And now I'm going to give you an idea about the importance of these SDHs in animals. If you have a polyol pathway, which is SDH is appearing as an intermediate enzyme, which converts glucose into fructose via sorbitol. So in this pathway, if you make fructose, it helps the cells to maintain its redox balance. And at the same time, if you find that the sorbitol is excessively accumulated in the cells, you would see osmotic stress as well as cellular damage. And if you are a diabetic individual, you would see that there are some implications taking place in your body because of this. For example, you could see cataract formation in your eyes and in problems in the neuron system, and as well as this kidney disease, which is caused by this cellular damage. And in plants, you can see the polyol pathway, which is controlled by both the aldose reductase and sorbitol dehydrogenase enzymes. And as we mentioned for animals, you can see that if this enzyme is active, you will see that plant cells can maintain the redox balance, and it's acting as an osmolyte, and it gives resistance to these plants during stressful conditions like droughts. And if you see excessive sorbitol accumulation in the cells, that would lead to osmotic stress and then leading to reduced growth and yield in the plants. So as this enzyme is important for biological systems, there are some industrial applications of this enzyme that has been currently focused on. Well, one is production of chiral alcohols, where these SDHs are acting as an intermediate enzyme. Another approach is production of nutrient supplements, such as vitamin C, and also making rare sugars, which is, you may have found that rare sugars like tagatose and allulose are used as artificial sweeteners. So therefore, there are several applications of SDH in industry. Next, I'm going to introduce the origin of this enzyme that I'm going to talk about today. So this enzyme is originated from Caldiatribacterium inferamans SIUC1, which is isolated, identified, and described for the first time by Dr. Scott Hamilton Brehm. And so this is a subsurface bacterium. It's a strictly anaerobic thermophile. So as they are living in the subsurface environments, they may produce these extremozymes, I would say, the enzymes that can thrive under harsh conditions like higher temperature. So So these extremozymes are important. Based on this, we developed a hypothesis for this study. That is, we hypothesized that STH-SIUC1, which is coming from a subsurface bacterium, has industrially beneficial characteristics such as higher thermostability and can be used for bioindustry applications. So to study this hypothesis, we came up with a general objective for the study. So our objective was to get an in-depth understanding of structural as well as functional properties of this STH-SIUC1, which is coming from this novel, strictly anaerobic, thermophilic subsurface bacterium, Caldiatribacterium, inferamans, SIUC1, to use it in bioindustry. And we also have set some specific objectives to achieve our goals in this study. Basically, I have categorized them into 3 sections. One is expressing this protein heterologously and studying its phenotype characterization using a specific suitable host. And after that, studying the biochemical characteristics and also using in silico approaches to explain the functional mechanism of this enzyme. So when I go through the presentation, we will get to know what are these specific set of objectives. And first of all, when we had the amino acid sequence for this enzyme, we needed to do a phylogenetic tree analysis. So this amino acid sequence included about 348 amino acids, and we did a homology search in Protein Data Bank and NCBI protein blast search. Here, after finding the similar sequences for this enzyme, we constructed a phylogenetic tree using Megalavan. And here I have included the phylogenetic tree that we constructed. And there are 4 different groups in this tree. That in the group A, you can find sorbitol dehydrogenases from human and plants. And in the group B, you can find STH-SIUC1, which is enclosed with some other members of Atribacteria or P9 phylum. Here I have included the group B, where you can find the closest relative to STH-SIUC1 was this NADP plus dependent alcohol dehydrogenase, which is described from Candidatus Caldiatribacterium californians. So this bacterium is not cultured yet. Its identification is basically from metagenomic analysis. So next, I'm moving on to talk about the biochemical characteristics of this enzyme. First of all, we needed to have the purified protein to do this. So to purify the protein, we constructed this plasmid, which is P-late31 STH-SIUC1 using LIC cloning. And after that, we transformed this plasmid into E. coli BL21. And in the protein production experiment, we used E. coli BL21 P-late31 STH-SIUC1. And we had to grow it in a nutrition-rich LV medium and then transfer into TB. And we had to induce its protein production by adding IPTG. And we used a nickel his gravitrap column to purify the protein at the end of the process. So this is the SDH page for the purified STH-SIUC1 protein. As you can see here, this is the band for this protein, which is about 37.8 kilodalton in size. And then the next step was to find out the oligomeric state of this protein. That is the native structure, native state of this protein. So to do that, we run a native page, but our native page was not that conclusive about the structure of oligomeric state of this protein. So therefore, we could see that it's lying between 130 and 250 kilodalton. To confirm this further, we had to go to Dr. Divya Prakash from chemistry department to do a FPLC analysis, which is size exclusion chromatography. So by that analysis, we found that size is about 25 kilodalton, which is indicating that it's going to be a hexamer. However, we have to confirm this data further because we could find that in our protein sample, there were some impurities also. So we have purified the protein sample further and run these analyses again to confirm that this is a hexamer. So basically, when you talk about the STHs produced by bacteria, they have been dimers, and human sorbitol is it's a tetramer. So there are some multiple proteins have been recorded for STHs, but this is turning out to be a hexamer. And after having the purified protein, we needed to find out what are the substrates that this enzyme can catalyze. And at the same time, we had to confirm what are the cofactors needed for this enzyme's activity. So to do that, we conducted a series of enzyme assays at 37 Celsius and pH 7 using different substrates. And for each substrate, we studied what is the cofactor which is catalyzing, which is helping this enzyme to catalyze the reaction. So according to that results, we can see here that sorbitol, xylitol, inositol, those are sugar alcohols, that the conversion of these sugar alcohols could be catalyzed by this enzyme in the presence of NAD plus as a cofactor. And here I have 2,3-butanediol, 1,4-butanediol and glycerol. Those are diols and triols. And they also needed NAD plus in the reaction mixture for their conversion. And here, this is an aldehyde, which is glycol aldehyde, that can be converted into ethylene glycol by NAD in the presence of NAD plus in the reaction mixture. Those two are fructose and sorbose. Those are ketoses. They needed NADH in the reaction mixture for its conversion. So these results indicated that STH-SiUC1 is a promiscuous enzyme in terms of its ability to do oxidation and reduction reactions, where it converts aldehydes, sugar alcohols, diols and triols, and ketoses into their products. And here we did the rate calculation for these conversions. We observed that higher rate was observed in fructose, sorbitol, xylitol, and sorbose conversion. And this table indicates the cofactors and the substrates that reacted with the enzyme. So according to this data, it says that STH-SiUC1 is an NAD plus NADH dependent enzyme. And to further confirm this data, we did the kinetic property calculation. So according to the kinetic properties, we used the substrate sorbitol, xylitol, sorbose, fructose, and glycol aldehyde that showed promising conversion in the earlier experiment. So according to that, we could see that sorbitol and fructose has a higher affinity for this enzyme. And then we needed to further confirm that this enzyme is capable of carrying out the sorbitol conversion in a reversible manner. For that, we did an HPLC analysis of our reaction mixture. Here we have the first chromatogram, which shows us that sorbitol is converted into fructose. And here we can see the fructose key. And in the second one, it is the reaction that fructose is converted into sorbitol, where we can see the sorbitol formation using HPLC. So this confirms that STH-SiUC1 catalyzes both the forward and reverse reaction of sorbitol conversion. And next, we wanted to study effect of several physicochemical parameters on the STH-SiUC1 activity. First of all, we determined how does the metal ligands can affect the enzyme's activity. So for that, we choose zinc, calcium, magnesium, copper, manganese, and ferrous. And here in the enzymatic reaction mixture, you have sorbitol and one of these metal ions at pH 7 reacting. And here we have the rate calculation for this conversion. And we observe that highest rate of NADH production by converting NAD plus into NADH was observed when zinc 2 plus is added into the reaction mixture. So this shows us that STH-SiUC1 prefers zinc for its function. However, we could see that magnesium 2 plus as well as manganese 2 plus also has the ability to accelerate this reaction. However, we didn't observe any significant difference between calcium, ferrous, and copper 2 plus effect in this enzyme's activity. And we also calculated kinetic parameters to confirm the previous data. Here we can see that zinc 2 plus has the highest conversion rates when it is added into the reaction mixture, where the STH is converting sorbitol into fractals. And our next experiment was to determine effect of pH on STH-SiUC1 activity. So here we used the sorbitol conversion reaction, and we provided different pH into the reaction mixture using different buffers, starting from pH 3, around 3.8 pH up to about 10.6 pH. So we could see that the highest conversion or the highest change in the absorbance due to the NAD plus conversion into NADH was observed at higher pH levels. So we did a kinetic property calculation for this experiment also, that we could see that when the pH is lower in the acidic level, you couldn't see the enzyme is active, converting the substrate into its products. However, when you use the alkaline conditions, you could see that enzyme prefers alkaline conditions in converting its substrates. And next step was to study the effect of different temperature on these enzymes' activity. So we did this experiment for both sorbitol conversion as well as fructose conversion. And here we selected a range of temperatures starting from 30 up to about 85 to study how does the OD340, which is respective conversion of NAD plus to NADH or NADH into NAD plus, will fluctuate depending on the temperature provided into the system. So then we conducted the rate calculation for both the reactions. So we could see that this is the sorbitol conversion in different temperatures. So we could see that there was a gradual increase of sorbitol conversion with temperature starting from 30 to 60. After that, we couldn't see this. We could see that there were ups and downs in the rate of conversion. So we concluded that 60 Celsius is the optimal temperature for this reaction. However, enzyme was active at 85 Celsius, which is the optimum temperature for the enzyme's activity. And when we consider the fructose conversion, we could see that fructose conversion was taking place at a higher rate when the temperature was increasing, depending on the temperatures provided into the system. And in the next step, we wanted to study the thermal stability of STH-SiUC1, because now we understood that the enzyme's highest, the highest temperature that the enzyme can work was 85. So we wanted to find out for how long that the enzyme would stay active at 85 Celsius temperature. So to do this, we used two different conditions. One is we pre-incubated the enzyme at 85, adding zinc into it. And the other condition was the pre-incubation of enzyme at 85 without adding zinc into it. And after that, after pre-incubation, we got the aliquots of enzyme to run the enzyme assay for both fructose and sorbitol at 30 Celsius temperature. So what we observe here is when you have zinc 2 plus added into the reaction mixture after in the pre-incubated enzyme, if you have zinc 2 plus added, we observe an elongation or extension of thermal denaturation point for both the reactions. If you talk about sorbitol, without zinc, it was terminated at 12 minutes. And when the zinc was there, the thermal denaturation point was increased up to 20 minutes. And in the fructose reaction, we observed without zinc, it would terminate the reaction at 16 minutes. And when the zinc is present in the pre-incubated enzyme, the thermal denaturation point was increased up to 25 minutes. So here we observed about 66% of thermal denaturation point increment in sorbitol reaction, while it was about 56 in fructose conversion. Here we tried to do a rate calculation for this previous experiment, that we could see that at the thermal denaturation point, there was an increment in the rate of conversion. So for example, in the fructose conversion, we could see this is the without zinc reaction, while this is the with zinc reaction, that we could see a rate increment at the thermal denaturation point. At this point, we don't know what is the exact mechanism that this is suggesting, that we are planning to study about this further. And then as we observe that in the presence or absence of zinc is affecting to the rate of this enzyme, we decided to synthesize the enzyme without adding zinc and with added zinc. So after synthesizing the protein under these two different conditions, we did a Western blot to study the two proteins are synthesized. And then we use these two enzymes, that enzyme synthesized with zinc added and without zinc added in the enzyme acids. So here I'm going to talk about the sorbitol conversion. As we observed, both the enzymes were converting sorbitol at the 30 Celsius temperature. So we could see that there was this increment of rate of conversion for the zinc enzyme synthesized with zinc, adding zinc. So that means, as we talked about earlier, the presence of zinc 2+ can increase the rate of this reaction. And what we observed when we pre-incubated the enzyme, the two enzymes and used in the enzyme assay, we could see that the enzyme with added zinc was still retaining the activity for sorbitol conversion, while we observed that enzyme without zinc had a loss of rate in the activity. So this could indicate us that the presence of zinc 2+ can affect both the structural and functional stability of this enzyme. And we did the experiment for fructose conversion. So we observed the same trend in the results at 30 Celsius temperature. And here, however, we observed higher rate for the enzyme synthesized with zinc than the enzyme synthesized without zinc. So the significant difference in the rate was observed when the enzyme was pre-incubated at 85 Celsius. So here we could see that enzyme synthesized with zinc 2+ showed higher activity, higher rate, and than the enzyme synthesized without zinc. Here, we could say that we have provided a temperature effect at 85 for the two enzymes here. So we could see that it affects both structural and functional stability, because this rate was less than the previous experiment when you use the enzyme without the pre-incubation at 85. However, the enzyme still managed to be active at 80 after the pre-incubation and still convert the substrate into its product. So based on this observation, we decided to look for the published literature that is talking about giving zinc 2+ a structural stability for SDH-like enzymes. So then we found that there is an enzyme reported, this is ketose reductase coming from silver-linked whitefly, that is suggesting the presence of a structural zinc binding site that provides the stability to the enzyme. So we did an alignment of our amino acid sequence from SDH-SIUC1 to the amino acid sequence of ketose reductase. So we observed that there are some conserved regions that can act as structural zinc binding site in both the enzymes. So depending on that observation, we came up with a hypothesis. We tell that is as the addition of zinc 2 plus increased the stability of STH-SiUC1 at higher temperature. Our hypothesis was that STH-SiUC1 has a structural zinc binding site. So to prove this further, we decided to mutate our STH-SiUC1 in the tentative structural zinc binding sites of the amino acid sequence. To do that, we used the Q5 mutagenesis technique to replace this structural zinc binding site is reported to have 4 cysteine residues, which we replaced with 4 serine residues using Q5 mutagenesis. And we constructed the same E. coli BA21 strain to do the protein purification. And here we have the Western blot for SDS page that shows us that the enzyme shows still the 37.8 kilodalton size. And this is our native page. Although it is not that conclusive, we could see that still the band is band representing this enzyme is lying between 130 to 250. That means it might indicate that still the structure or the oligomeric state of the protein is not changed. Therefore, we needed to find out whether we can see any activity change in the enzyme. For that, we first of all, we conducted thermal shift assay, where we can find out whether we could see any change in the melting temperature of these 3 enzymes. So the 3 enzymes are enzymes synthesized with adenosine, without adenosine, and SDH mutant. So based on the thermal shift assay, we observed that although the melting temperature for the enzyme synthesized with adenosine was about 92, and without zinc it showed 88 melting temperature, and when it is coming to the mutant, the melting temperature dropped about by about 14 Celsius, which is around 78. That means that the structure of the mutant has been affected by the mutation that we introduced into the amino acid sequence. So to further confirm this, we conducted the enzyme assay using both all the 3 enzymes. So here, this is the sorbitol conversion that we observed that the other 2 enzymes that we used as a control was giving the activity as we talked about previously, but we couldn't see that mutant was converting sorbitol at this condition. So that means after introducing this mutation into the tentative structural zinc binding site of the enzyme has lost its activity completely. Here is the fructose conversion. We observe the same thing, that now the mutant cannot convert fructose into its product. So it seems its catalytic activity is lost due to the mutation that we introduced in the enzyme. So to further confirm that the enzymes carry zinc, we got the help from chemistry department to do a ICP-MS analysis of zinc in these 3 enzymes. So according to that data, we observed that the highest zinc was present in the enzyme synthesized with zinc. We couldn't see any zinc in the enzyme without zinc, and the mutant contained some amount of zinc according to this analysis. However, this is our first analysis about this. We have to repeat this analysis to further confirm this observation. And that was about biochemical characterization of the enzyme. Then we wanted to study whether we can express this gene in a suitable host. So we chose Pseudomonas butida KT2040 as our host to express this gene. So we selected this bacterium because it is a robust, metabolically versatile bacterium, which is recognized as can thrive in harsh conditions and participate in production of a series of metabolites. So as we found, this is the best candidate for our work, we decided to integrate STH-SiUC1 into the genome of Pseudomonas butida KT2440. And after that, after the genome integration, we decided to do a RT-PCR analysis to check whether the protein is expressed or the mRNA expression we could detect mRNA expression in this strain. So according to the RT-PCR analysis, we could see that this gene is expressed in Pseudomonas butida KT2440. After that, we needed to confirm the phenotype of this organism. For that, first of all, we did a plate assay that using sorbitol as a sole carbon source in a minimal medium M9. So we observed that this strain could grow on sorbitol plates. So that suggests that this might have a sorbitol transporter that can take up sorbitol and funnel that sorbitol through a biochemical pathway. That might be the pathway like EMP pathway, where the sorbitol is funneled. So to further confirm this data, we conducted a tube assay where we used sorbitol as a sole carbon source. And we also needed to study the growth of these wild-type Pseudomonas butida KT2440, as well as the STH strain in glucose. And after that, we used sorbitol as a sole carbon for the growth of these two organisms. Although we observed that engineered strain was growing in sorbitol-containing medium, we couldn't see the wild type was growing there. So that means it is because of the expression of STH-SIUC1 in this particular bacterium. And we also did a sorbitol utilization experiment by HPLC to check whether this organism is converting sorbitol. So we could observe by HPLC that the sorbitol was converted within the time points that we did the assay. So if I give a summary of biochemical characters of this STH-SIUC1, we could say that it's a hexamer and have a higher affinity for sugar alcohols like sorbitol and xylitol, and ketoses like fructose and sorbose. And it's an NAD plus NADH dependent dehydrogenase, which prefers Zinc 2+, and it prefers alkaline pH conditions, and optimal temperature would be 60 and maximum temperature is 85, and has a structural zinc binding site, which would mutations in that structural zinc binding sites would lead to the losing of catalytic activity of the enzyme. And we have to study whether the substrate preference could be changed from sorbitol to fructose at higher pre-incubation temperatures like 85. And it was also possible for us to aerobically express this STH-SIUC1, which is coming from an anaerobic microorganism in E. coli and Pseudomonas butida KT2440. Those are aerobic microorganisms in terms of Pseudomonas butida KT2440. And the next section of our work was to use in silico modeling to describe the structure and function of this enzyme. To carry out these experiments, we actually this part of the study was led by Dr. Sandhya Jayasekara. So first of all, what we did is we predicted a 3D structure for the enzyme. Using the amino acid sequence, we used the SIS model software, which is a web-based software to predict models for the amino acid sequences. After predicting a 3D structure for the enzyme, we needed to locate the catalytic zinc binding site of the enzyme. So for that, we use the alignment of templates predicted by SIS model with the STH-SIUC1. So we could locate that the catalytic zinc binding site has cysteine, histidine, and glutamine residues conserved in its catalytic zinc binding site in all the templates, as well as in SIUC1 enzyme that we are studying in this work. And here is a comparison of catalytic zinc binding site of the templates as well as STH-SIUC1. So this is one template predicted for this STH-SIUC1, that is ketose reductase from silver whitefly. and this is human sorbitol dehydrogenase, we could see that the catalytic zinc binding site is composed of cysteine, histidine, and glutamate residues. So that was a common feature for all these three enzymes. So that means the catalytic zinc binding site is conserved in these enzymes. And this is a short video of catalytic zinc binding site of the enzyme CHSIUC1. When the catalytic zinc is coordinated into its active site, you could see these amino acid residues are coordinating and making a tetrahedral coordination in the catalytic zinc binding site. And then we needed to locate the structural zinc binding site. So I talked about this earlier in our experimental data. Here we have this structural zinc binding site, which is conserved with 4 cysteine residues. And here we have a comparison of structural zinc binding site in the templates. So this is the ketose reductase, which carries a structural zinc binding site. And this is STH-SiUC1 structural zinc binding site. And here we have 1PL6. However, 1PL6 means the human sorbitol dehydrogenase, but it doesn't carry a structural zinc binding site. There are some cysteine residues conserved, but the total structural zinc binding site is not conserved in human sorbitol dehydrogenase, but it is present in STH-SiUC1. And this is a preliminary RMSD calculation to study how does the stability of structural zinc binding site and how does the stability changes when both the structural zinc as well as active site binding zinc is there. You can see this is the responsible graph for active site zinc. In the presence of active site zinc, the stability of the enzyme is lower. But when you have both the structural zinc as well as active zinc bound to the active site as well as structural zinc binding site, the stability of the enzyme is increased, which further supports our previous experimental data that showed us that the enzyme structural zinc binding site has functional as well as structural stability given to the enzyme. And then we needed to find out the cofactor binding site of the enzyme. And we also needed to confirm that this enzyme requires NAD+, but not the NADP+. So that fact is determined by the presence of aspartate or alanine in its active site vicinity. So here we have 1E3J, that means the ketose reductase from silverleaf whitefly, which needs NAD+ as a cofactor. But the other enzymes, including human sorbitol dehydrogenase and STH-SiUC1, needs NAD+ in the reaction, which is confirmed by the presence of an aspartate residue in the enzyme amino acid sequence. So the cofactor binding site of STH-SiUC1 contained arginine, leucine, isoleucine, as well as tyrosine in the vicinity where NAD+ was binding. And this video shows the binding of NAD+ into the active site of the enzyme. You can see here, this is the zinc, which is coordinating the active site. And here we have the NAD+ bound to the catalytic site of the enzyme. And here, after confirming the structural zinc binding site and catalytic zinc binding site and the binding of NAD+ into the enzyme, we wanted to try and find out what is the mechanism of dehydrogenase of this STH-SiUC1. So here we can see that this is the catalytic zinc binding site. And here we have, this is representing the sorbitol, which is the substrate for this enzyme. And this is NAD+, which is the cofactor. And when all these ligands are bound to the active site of the enzyme can start the catalyzing the reaction. And the first step would be to remove a hydride atom from the substrate and give that hydride to the NAD plus, which is in the vicinity, and convert NAD plus into NADH. And at the same time, because of this, sorbitol would be converted into fructose. So this is a summary of in silico characterization of the enzyme. So we have predicted a 3D model for STH-SiUC1, and we have described the catalytic zinc binding site as well as structural zinc binding site and the coenzyme or cofactor binding site. And we also proposed a functional mechanism for STH-SiUC1 activity. And here I have the conclusions of this study. For the first one is like STH-SiUC1 is an NAD plus NADH dependent dehydrogenase. It's a thermostable enzyme with two sites like structural zinc binding site as well as catalytic zinc binding site. And these two sites provide both the functional as well as structural stability for the enzyme. And pre-incubated STH-SiUC1 at 85 Celsius, we think that it is a substrate preference from fructose to sorbitol that we have to further study. And in combination with in silico modeling as well as biochemical approaches, we have described a functional mechanism for this enzyme. So in future direction, first of all, we have to get the crystal structure of STH-SiUC1 because it would help us to confirm most of these properties that we came up with in the study. And we also need to study whether the enzyme can switch its substrate preference at higher temperature levels like 85 Celsius. And also we need to do a full phenotypic characterization of Pseudomonas mutata KT2440 STH-SiUC1 to study its potential role of STH-SiUC1 in this organism. And there would be potential applications of this strain in rare sugar production and chiral alcohol production that we plan to study in the future. And we also observed that this enzyme can react with glycolaldehyde like aldehyde, which are toxic molecules. So we have to further study whether we can use this STH-SiUC1 for alleviating toxic molecules like these aldehydes. And also we observed that this enzyme has a structural zinc binding site, which provides thermal stability to the enzyme. So if we can use this as a model to introduce thermal stability to mesophilic enzymes and improve their activity, that would be an advantage that we are planning to study further. And also by further MD simulations and in silico related studies, we would be able to suggest beneficial mutations to further enhance activity of this enzyme. So those are our future directions. These are some of the publications that we did during this work. And these are conference proceedings that I presented my work at different conferences. And here, after completing this, I would like to thank, first of all, Dr. Sandhya Jayasekara for being an excellent advisor. And my colleagues in the CIMIC lab, Lakshika, Bhagya, Sandipti, we are not just lab mates, we are like, they are giving us giving a lot of emotional support during our work. And the undergraduates and training students work with me, Abigail, Rion, Lily, and Isabel. They gave a lot of support to do this work. And my thesis committee members, Dr. Derek Fisher, Dr. Bethany Rader, and Dr. Sandhya Jayasekara, their guidance and support in this work has been fabulous. And our collaborators, Dr. Siva, Dr. Scott, and Dr. Divya. So their support in this work was tremendous. And I would also like to thank faculty and graduate students in the microbiology program at SIU, because we have got a lot of help from them. when we were doing our research in the department. So I would also like to thank our funding, because that is where we get financial support to do this work. And thank you so much.